Our next speaker is Dr. Antoine Roux. He is a principal scientist at Calico Life Sciences, where he works on both exploratory research and translational projects related to aging and age-related diseases. His interest in aging biology originated from his PhD project at the University of Montreal, Quebec, where he pioneered aging biology in the fish and yeast. He then continued studying aging in the nematode C. elegans during his postdoc at UCSF with Dr. Cynthia Kenyon. There he discovered that aging could naturally occur and be reversed at the embryonic stage. More recently, at Calico, he generated, with the help of David Kelly's team, the first complete cell atlas of an aging multicellular organism using, again, C. elegans.
Hi, everyone. Okay, thank you very much, everyone, to be here. I'd like to give a big thank you to the organizers, particularly Christine here. So you probably heard about BAM, which happened this year, and I think this is the perfect addition to BAM, to give a tribute to the private sector. So I hope that it will last for a long time. And yeah. So I work at Calico. I've been there from almost the very beginning. And oh wow, we have a cursor. Yeah. So at Calico, we really have we're mostly involved in translational biology and try to generate targets and new therapies and to bring to the clinic, particularly in collaboration with AbbVie, our sister company. So things are doing well. I'm sorry we don't show much, but we also have exploration science going on there, and so that's what I'm going to be presenting today. So this work has been done in collaboration with the Kenyon lab, the Kelly lab, and the Hendrickson lab. And I want particularly to give the credit to Han Yuan as well, who has been equally contributing to that project with me. So you've probably heard about C. elegans, but they are these tiny little 1 millimeter long worms, and they really help biologists a lot, particularly to study aging. So we've decided to start a project to kind of generate a single cell atlas, meaning looking at the gene expression of at the cellular level in the entire animal during the entire course of their lifespan. And this approach is kind of one of the favorite or very popular approach today in the sector, in actually every sector of biology. And really there are still important questions to answer here, like how this kind of data can be used, how valid these data are to measure aging. So I'm not going to so it's kind of sort of a clock, even though I'm not going to be talking about clocks, but really we kind of ask in every single cell of the animal, how do they age, and try to extract as much information as we can to also answer this question that is still widely open: what is aging? So using C. elegans is particularly advantageous for us because it's an animal that has been studied very, very intensively over the years, particularly from this guy and then pioneer who has described every single cell of the animal and made this kind of map. So we know every cell of the animal, and we know from the whole lineage, from like the egg to the adulthood. And so that's really convenient for us for reasons you're going to understand on the next slide. So he found that the animal contained about 1,000 cells plus germ cells, and there's around 220 different cell types. So what we've done is we've used a quite classic approach called single cell sequencing through the technology called Chromium, 10x Chromium. And what we've done is we've used very large populations of worms at every age to extract cells, like you can see here. And we have all sorts of cells, and then we sequence them at every age. Okay. And then we get this plot that Eric has already described called a UMAP. So pretty much what you see here is like every little dot is a cell, and we project these cells. So based on their expression, on how their gene functionate, you will see they will be form those groups. So every group, we call them clusters. Every group represents a cell type here. And so what we don't know we need to do at this point is to what we call annotate. So identify who is who. So we need to put a name on each of these groups. And so we've done that, and we found that we have like 124 cluster of neurons, maybe around 20 cluster for muscles, etc., etc., and like that, and even glia and then we've also used microscopy to verify the validity of our data. So we then take a gene that is specific to one cluster, like this little one down there, 96. And when we look in worms, we associate the gene to a fluorescent protein, and then it went to the place that we described as M5 neurons. So that means our annotation is valid, and we've done that for around 20 different cell types. So in the literature today, you'll find a lot of atlas for aging, and one of the most famous one is called the Tabula Memory Senis from the BioHub. So this is a consortium of over 10 labs who has extracted sample from mice tissues and then run the similar approach that I just explained for C. elegans. But really, it's not very fair to compare their data to ours. What actually they've done is that they've run organs separately, right? And that's really, that's the best they could do. So they run each organ separately, and they got this UMAP. But for us, because C. elegans is small, we could do it all at once for the entire organism and in the same lab. So what this actually gave us is like a higher resolution. So we can reduce the what we call the batch effect, right? The covariates that are due to non-biological factors, like temperature in the room, like who extract and who run the protocol, and all of this usually will change the data a little bit. So the cool thing for us is that because we can use this much simpler animal, we actually increase the resolution of our data set and reduce the batch effect. So for example, here is the same UMAP where now we colored by tissue type. As you see, mostly we have single neurons, and we actually resolve neurons by the class of them. So for example, the AIM neuron, there's only 2 cells in the worm that are the AIM neurons in one worm. And if we compare that to what they got in the Tabula Memory Senis, they only have 1 cluster for the whole neurons, entire neuronal population of mice. And you can imagine that, of course, in mice there is many, many classes of neurons, right? So, and again, today you'll find data set of the brain. There's one that is actually published as a preprint from the Tony Viscore lab that did resolve neuron to the same extent as us in mice, but they only get data for the brain only. So they focused on one organ and they compare it to the same. So what we have now is that we have an aging data set for the entire animal, and we can actually make comparisons between organs and cell types. And so what we have is that, so for each cluster, we have an age, right? We have cells that are from day 1, and then day 3, day 5, day 8. So we resolved age very well, which really was not so clean in the Tabula Memory Senis for neuron again. They didn't cluster separately age very often in their data set. So now what this data set really taught us. So what we see here is that every single gene of the C. elegans, and here you have all the tissues. And in blue means the gene tends to be downregulated with age, and in red it's upregulated with age. And really the first thing that you observe is that most of the genes are kind of this little islet. We don't see a lot of long bands here, blue, or there's a few of them, but not that many. And what that means is that it means that mostly, and that's something that Eric has touched upon earlier, mostly we see heterogeneity. We see tissue change in their own trajectory. So now when you ask what are the genes that are the most consistent, yet there's a few of them that consistently age in the animal. And what we found is that the ones that go up tend to be chaperones in the cytoplasm, and the ones that go down tend to be either involved in the ER-UPR, meaning the unfold protein response in the endoplasmic reticulum, a subset of the cell, or involved in the metabolism. So now we kind of run a similar question than this one, like really, what is the nature of aging in every cell type? But now we ask how the whole gene set, so group of genes that describe functions, so we call them the Go terms, how do these functions change with age in every cell types? And so what we found is that the most consistent one is really the downregulation of carbon metabolism and energy metabolism. It involves a lot of genes implicated in mitochondrial respiration, for example. Then we see, and that's here, right? So you see a lot of blue, and that's pretty consistent. Then we see some that also seems to be shared in many clusters, but not universal, like the ER, UPR, here. See that, and it's even more down in the neuron than the other cell types, for example. And then we see a lot of cell-specific things, like collagen is really downregulated, but in the clusters that are mostly kind of similar to the skin. So it's really function-specific and, of course, tissue-specific. And then we see some actually pretty surprising things, like this, just thinking, time, okay. So ribosomal genes. So the ribosome is encoded by some genes that forms the protein part of the ribosome. And what we found, and this also has been kind of suggested and observed also in mammals, and not very commented upon, but I think our data show that it's really real, that the ribosomal encoded gene are highly regulated, highly going up or highly going down. Here's one gene in one cluster and the same gene in another cluster here. You see that really the gene go up or go down very significantly in different tissues, and that can be non-neuronal or neuronal either. So we don't really know if this somehow regulate the rate of aging in the cell types, but our lab has published a while ago that if you do mutations in those genes, you do slow down longevity, slow down aging, sorry. So, so there's a possibility that they are important that will, I mean, and you know, translation is important, so that this actually regulate the rate of aging. But in our data set, we haven't been able to find any kind of anything to conclude about that yet. Okay, so now we've tried to ask, instead of really what's the nature of aging in every cell types, we try to quantify the rate of aging in every cell types, and then compare it with one another. So what you can, I'm going to show you here two different methods. So one is called MMD, maximum mean discrepancy, and that's kind of a fancy way of just asking if you have a big change of the overall transcription, every genes between young and old. So you know how far apart these distributions are. And so we do that for every cell type. So every cell types are represented by dots here. And what you see is you have huge variability. So neurons, within neurons or within muscle cell types. But we see that, for example, neurons tend to age faster than muscles, for example. And then we also use another matrix called increased cell variability, and this one kind of measures the spread of the cells in the UMAP. So if the cells get more different with time, and that's above this line here, so each dot here, they are very close, so they form a line, but actually they are all the cell types. And you see that if they are above this line here, that means the cell within these cell types tend to look more different with time, right? So there is more kind of noise, more chaos within the cells maybe, and the cells just spread. And then what we were surprised to find is that there's a subset of cells where that actually gets more alike with time. So really the increased noise and increased variability, which people use sometimes to quantify aging, is not a necessity. Like sometimes cells can actually be more similar. And now what we've done, we use the beauty of the worm. The worm are transparent. They live not very long, like 2-3 weeks. And so we took tissues that were measured by expression data to age faster than others, like this blue here, celomocytes, or red here, which is a SIM cell. And it's also the case for cell-to-cell variability. And we asked how does tissue look during aging in the worm. So in these pictures that are not of great quality, sorry, you see that in green we mark the mitochondria, and we know that mitochondria morphology change with age, and we compared this, over time in the animal. In the same animal, we compared these two tissues, and we've done it for another pair of tissue, and we confirmed that really the mitochondria changed much more in the tissue that we predicted to live long, to age faster, right? So that confirming that using this approach is valid to measure aging. So in conclusion, we found that really cell types trajectories can very diverse in terms of aging, and that even worms cell types can suffer different stress, or they respond to different stress, like DNA damage in some types. Sometimes ER, UPR going down, and sometimes we see more like cytoplasmic chaperones going up. So suggesting that possibly they age in a different way. However, this seems to have at least some level of conservation, and that could be that maybe there is an underlying mechanism that will just be interpreted differently by the different translation programs. And so then we kind of validate the use of single-cell RNA-seq to measure aging. And all our data are available online on an interface that even if you don't know how to code, you can actually play with this data and maybe make discoveries. And okay, in 20 seconds, we've also then done an analysis of every transcription factors in all tissues during the lifespan of the worm. And we found pretty cool things that the transcription factors that change the most consistently within all our tissues, they tend to go up first. And those transcription factors are actually very canonical, known, and published pathways related to longevity. So for example, you see like DAF16, skin1, or this autophagy regulation, TFEB homologue. So that's pretty interesting because it means that maybe others here are also regulating longevity and have not been found yet. So actually, I run a screen based on that, and I isolated a few new transcription factors regulating longevity, like GI3 here, that regulate both the survival and the decline of speed of the worms, the health. So that's it for today. Thank you very much for your attention. Yeah, you're probably aware that there's something going on outside here that. I don't know if Spain won, by the way. But so thank you for your attention and your enthusiasm. I wish people were that excited for science, but it's soccer players, but that's okay. I'm not mad at all. Okay.
Thank you. Fantastic work. Thank you.
Thank you, Gordon.
I'm wondering about interventions, compounds, drug-like molecules that extend lifespan in a worm. And one of the big questions is, are they doing it in the same way? Are they doing it in different ways? Have you started looking at different compound lifespan extensions and looking at the profiles that result from that?
So unfortunately not. We have done an attempt for DAF2. Unfortunately, we've used the auxin method, you probably heard, like to turn down DAF2, and unfortunately, I need to grow the worm in like thousands of very large population, and I mean, long story, but at the end, we were not satisfied by our increased longevity in those population. It works great, but we decreased the dose of auxin too much, so then we did not actually work on this data set because the worms were not long-lived enough. But that's it. We haven't done anything else. Yeah.
Allez les bleus.
It can be a question about soccer too.